Good morning. Welcome to PCAP's Native Prairie Speaker Series. My name is Caitlin Rose Seiler, and I am the Stewardship Coordinator with Saskatchewan Prairie Conservation Action Plan, or PCAP. Today, James Paterson from Ducks Unlimited Canada will be speaking about supporting conservation decisions with the Biodiversity Mapping and Assessment tool. Before we get started, I'd like to respectfully acknowledge that we are on the traditional territories of many Indigenous nations and communities, past and present. For millennia, they have worked to protect these landscapes and the life these areas sustain. I'd like to thank these original caretakers and acknowledge the ongoing work and presence of Indigenous peoples in Canada today. I'm delighted to let everyone know that we have an awesome lineup of webinars happening this winter. Topics include grasslands birds, bats in the Cypress Hills, long-billed curlews, as well as swift fox. So check out the PCAP website to register for the upcoming webinars. A reminder to our listeners out there, you'll be muted for the duration of the presentation. But if you have any questions during the presentation, just type it into the Q&A section of the webinar dashboard at any time during the presentation, but questions will be answered towards the end of the webinar. And you're also able to upvote other people's questions if you have the same one. I'd like to take a moment to note that financial support for today's webinar is provided by our presenting sponsors, Canadian Forage and Grasslands Association, North American Helium, Pembina, Saskatchewan Cattle Association, and SaskTel. Our supporting sponsors are Innovation Credit Union and Rancher Stewardship Alliance Inc. In-kind support has been provided by Ducks Unlimited Canada. And without further ado, I am pleased to introduce today's presenter. James Paterson is a research scientist in biodiversity at the Institute for Wetland and Waterfowl Research, which is the science arm of Ducks Unlimited Canada. He completed his Ph.D. at the University of Ottawa, his Master's of Science at Laurentian University, and his Bachelor's of Science in Zoology at the University of Guelph. Prior to joining Ducks Unlimited Canada, he was a Liber Ero Fellow at Trent University. The goal of his research program is to explain spatial and temporal variation in biodiversity and to improve the application of conservation interventions such as habitat protection and restoration. So with that, I'll pass it over to you, James.
Thank you. Caitlin, can you just confirm that you see the slides okay?
Yes, looks perfect.
Okay, perfect. Thank you everyone for coming. My name is James. I'm going to be talking about our biodiversity mapping and assessment tool and how it can be used to support conservation decisions in Prairie, Canada. Before we begin, I want to acknowledge that I'm presenting from Okamic Marsh, just north of Winnipeg on Treaty 1 territory. For those unfamiliar, Ducks Unlimited Canada is a habitat conservation organization with a mission to conserve, restore, and manage wetlands and associated habitats for the benefits of waterfowl, wildlife, and people. And since 1938, the organization has conserved more than 2.51 million hectares of habitat through more than 13,000 habitat projects. As a science-based organization, Ducks Unlimited Canada uses decision support tools and the best available science to help guide where work is done and how projects are delivered in order to maximize our investment impact for our mission. And so I'm going to talk about one of the projects in which we're looking at how these habitat conservation projects are benefiting biodiversity. I also want to acknowledge my co-authors and collaborators on this project, including Lauren Page, Jim DeVries, and Ash. To set the stage for why we started this work, currently global biodiversity is declining. The most recent global assessment report on biodiversity and ecosystem services by the Intergovernmental Science Policy Platform on Biodiversity Ecosystem Services, or IPBES, stated very clearly that biodiversity is declining faster now than at any other time in human history. In that report, they identified what the major drivers of biodiversity loss are, but the single largest direct driver is changes to land and sea use. And so in order to address this large single driver, society needs tools to be able to estimate how biodiversity is affected by land cover change. And so we wanted to take our small piece of this challenge and to build a tool to predict biodiversity based on land cover. This is a way where we can relate which species occur where to changes on the landscape and the different habitat types that occur. And our tool really works on two simple concepts. The first is that habitat loss and landscape simplification reduces biodiversity. As habitats disappear, so too do the species that rely on those specific habitat types. On the flip side, we know that habitat restoration, when done in the right context and location and places, can provide opportunity for biodiversity to return once those habitats are back on the landscape. And really, the models that we're building are just trying to estimate the shape and the nature of the relationships between all of these different species and the habitat types on which they rely. The work today I'm going to talk about is based on the prairies, but we also have similar work in progress for Eastern Canada south of the boreal forest. And so we chose the prairies because it represents an interesting overlap between an area that has high national biodiversity, but also is a working landscape containing most of Canada's arable land. And so this intersection between being very important for biodiversity, but also for growing food is one opportunity for us to find the best places and opportunities to be able to conserve biodiversity while maintaining these important features for humans. The geographic scope outlined in that red polygon is the Prairie's ecozone, which covers the southern portions of Alberta, Saskatchewan, and Manitoba. And the tool includes data and predictions on all 329 species of amphibians, birds, mammals, and reptiles which live in this area. The spatial scale which the tool works is on a quarter section or a half-mile by half-mile parcel of land. That decision to have that be the spatial scale on which we make predictions is driven less by biology and more about management decisions. This is the size of land that's typically used for land management and buying and selling land. And so it is useful to be able to frame our predictions at a scale that conservation decision makers are using. And so the tool takes broadly three types of data: information about species observations, where species occur, information about habitat, and then also data on climate. We know that our focus is on how species respond to land cover, but that climate is an important forcing factor on suitability of an area for different species. We combine those different data types to build species distribution models for the hundreds of species that occur in our study area. And when we stack those predictions together, we can get a picture of how biodiversity varies through space and what are the main drivers of those differences. For each of those buckets, we're using the best available data. So for species observations, which included more than 1.2 million observations, we used a combination of both publicly available data sources, such as the Global Biodiversity Information Facility, iNaturalist, and North American Breeding Bird Survey. But we recognize that especially for rare and endangered species, those data sources underrepresent those species. And so we also have data sharing agreements with the conservation data centers in Alberta, Saskatchewan, and Manitoba, which are the agencies responsible for tracking rare and endangered species. For habitat data, we're using Agriculture and Agri-Food Canada's Land Use Time Series, which has a land cover product at 30-meter resolution. And for climate, we used WorldClim. Species distribution models often have information about local climate, elevation, and soil characteristics, and we built all of those into predicting where species occur. But we were most interested in how species respond to land cover, and so we also included variables that describe the amount of habitat within a field, the amount of habitat within a larger landscape, because species respond to habitat at different scales. For example, if you think about how a frog might respond to habitat compared to a white-tailed deer, larger animals typically are responding at a much larger scale. We also included predictors based on habitat patch size, because many species are sensitive and require continuous patches of certain habitat types, for example, native grass specialists. Now, there's many different land cover types within this study area, but we simplified that to focus on the variables we thought are driving the most variation in the system, including wetlands and open water, grasslands, cropland, forest, and then human settlement. Now, without going into a deep rabbit hole on the exact statistical methods, I'm going to walk through an example of how we use this data to form these models. So for an example species like the Great Plains toad, we want to estimate how the likelihood that the species occurs at any site is related to a number of variables, including, for example, the amount of wetlands within a field. We want to draw what that green curve looks like. To do that, we take the characteristics of places where the species occurs, here represented by green dots, and we compare it to a large number of background points within the species range. Then the statistical model is able to distinguish what is the difference between a background point and the exact species presence points. We get a map of species habitat suitability, and then when we stack that across all of our different species, we can get a picture of biodiversity variation or the number of species that occur at each site. This is beneficial for conservation planning because it allows us to understand where to work to be able to target specific species or species groups, and also is beneficial for reporting after we're delivering a project to understand what species are benefiting from an individual project. To get a picture of what this looks like, we're going to zoom in on two fields that are separated by less than 3 kilometers. And so on the field on the left, outlined in yellow, is a crop field that has some remaining forest and wetland patches. In the field on the right, outlined in white, there's almost no natural vegetation and it's just cropland. And you can see that even though they're very close in geographic space, the climates are functionally the same, have very different number of species predicted to occur there based on the type and the amount of different habitats. And so this is useful for understanding which species are benefiting from, for example, protecting or restoring habitat on these different fields. So I ran through an example of how our spatial predictions of biodiversity, where different species occur, might be useful for planning and reporting on conservation action. I'm going to run through now an example of how we can use this tool to estimate the benefits of a conservation action. So we were interested in the question of how much conserving small patches of habitat might be benefiting biodiversity. In much of the prairies, conservation action work occurs on small patches that are embedded within a larger agricultural context. And conservation easements, which are a voluntary legal agreement between a landowner and a conservation organization, such as Ducks Unlimited Canada, to protect the natural assets on that property. For example, protecting wetlands from drainage or grasslands from being broken. We wanted to know how much an effect these are having for local biodiversity. And so we compared the tool's predictions of which species occur at these sites to nearby unprotected land to get a sense of, are they working in the way we think? So on the vertical axis, I'm showing the predicted number of species in the green for conservation easements and in the purple for sites that have no protection. Each hollow dot represents one site, and the filled in color dots represent the group average, and the vertical bars around those colored points is the uncertainty around that average. And so I'm looking at this plot, there's kind of two big patterns I want you to see. The first is that there's lots of variation. Some sites have very few species and some have a lot. But the other major pattern is that on average, when you're comparing the paired sites, which are connected by gray lines, the conservation easement sites have significantly more species than unprotected sites, and that difference is fairly large. We looked at this further to understand what is the mechanism for these sites having more species, and it's driven by the type of habitat there. Here I'm showing the same plot, but now on the vertical axis, it's the proportion of natural habitat within that field. And you can see a similar pattern where there's much more natural habitat on the easement sites compared to the no protection sites. In other words, this analysis suggests that conserving small patches of natural habitat within working landscapes benefits biodiversity. The magnitude of that effect is fairly large, and it's driven by those sites conserving more natural habitat than the nearby unprotected sites. And so we can use similar analyses to look at other types of conservation action that are conserving or restoring habitat. But I'm going to walk through another example that we can use these models to ask questions about what's driving variation in the system. And so we can do this by running scenarios where we simulate habitat change. And we can take a natural habitat that's full of grasslands and wetlands and forests in a organization that we think is typical of that system. And then we progressively convert more of that land to a different agricultural cover type. And so we can do this for the main agriculture land cover types in the prairies. The first one we considered was conversion of natural habitat to annual cropland. And so the first take home from this was that the proportion of annual cropland was the single biggest driver of local biodiversity loss. Here I'm showing on the vertical axis species richness as the percentage of maximum. And so 100% would be the most species that would occur at that site when you have all natural habitats. And on the horizontal axis, it's the amount of annual cropland sliding on the left to no cropland to on the right corner to corner cropping. And so there's a very consistent pattern among total species richness where there's this decline as you convert more land. And that pattern is consistent within each of the different groups we looked at: amphibians, birds, mammals, and reptiles. But we can use these scenarios to ask questions about what happens if we set aside different amounts of natural habitat. And so instead of having corner to corner cropping, what if we set aside 20% of natural habitats for conservation? And so in this scenario, conserving 20% of natural habitats conserves an average of 27% of the maximum number of species. And so this is a way of conserving relatively small patches of natural habitats, but still keeping almost 1 in 3 species that would have occurred there. Now, we did the same scenario with one of the other major agricultural land cover types, which is tame grass for forage or hay. So this is mixes of grass, alfalfa for haylands or for active pasture. So here the pattern is quite different. I'm showing the same vertical axis, the number of species richness as a percentage of maximum. But on the horizontal axis, now we're sliding from natural on the left to 100% tame grass mixture. And so here the decline is not as strong, and the decline is really strong only at the tail end. So if we take that same scenario where we set aside 20% of a field as natural habitat, in this case, conserving 20% of natural habitats conserves an average of 74% of the maximum number of species in this system. So by running scenarios like this, we can get a picture of how different program types or mixtures might have different consequences for the type and the number of species we're conserving in an area. Okay, so I ran through a few very quick examples with our biodiversity mapping and assessment tool to look at spatial predictions to understand which species benefit from program where. We looked at our conservation easement site to estimate the benefits of a specific conservation program to show that that program has both a large magnitude of effect for benefiting biodiversity, but also it's due to protecting natural habitats. And then we can run scenarios where we identify what are the major drivers of biodiversity variation in this system. Ultimately, all these applications are trying to take science-based solutions to help guide conservation so that we can have a larger positive impact on the landscape. Now, we use these tools internally at Ducks Unlimited Canada, but we also put together the information as a interactive mapping tool. And so there's a public version of our mapping tool. The QR code there will take you to the website that has more information about the data and allows you to search both by legal land description, but also any place within the prairies, and then get the prediction of the number of species that will occur there based on current land cover and climate data. There's also a data request option where you can request the underlying layer if you don't want to work in this specific interactive mapping tool. So those examples summarize our approach to our biodiversity mapping and assessment tool. The data analysis I talked about are included in an open access scientific peer-reviewed study, and then I've put the QR code for the public version biodiversity mapping assessment tool there as well. So I'm going to pause there and happy to take any questions.
Absolutely. There's a question about if you could show your screen again. Going back to the link or reference to the paper. It was earlier on in the presentation.
Yeah, I'll share my screen again with the QR codes, and I'll also drop the link in the chat. And if I can't get it in, then Caitlin can help me.
Perfect. Let's give you a minute to do that. Perfect. Yes, then people can pick up the QR code there. And then we have a question from Erica. She is wondering if you verify your models by ground truthing the sites.
I'm going to pull up a slide that describes our model verification. So we used kind of a tiered approach. So we basically used 3 ways to test and validate the tool. The first is in how we fit the model. Instead of fitting a single model for species, we used spatial K-folds that helped us estimate uncertainty for any individual species predictions. And that's what's typical with these species distribution models, is to use holdout cross-validation data. And so we did that where we basically are holding out 20% of the observations and sites, and then testing the accuracy with AUC, or area under the receiver operator curve, and then using a benchmark for identifying species with poorly performing models. But what's unique is that we also had a large independent data set. And so this was autonomous recording unit data from field sites at 192 wetlands in our study area. That none of that data was used to train any of these models. And then after we had the full models, we tested the predicted number of species with our models compared to field validated number of sites, and we found a strong correlation. Those sites aren't from every habitat type, so we were somewhat limited in the communities we can test. So those were from wetland field studies, but we found the number of wetland birds predicted by our tool was really strongly correlated with the number of wetland birds detected at our ARUs, which were deployed in wetlands at those sites. So we used a combination of how we fit it to be able to estimate uncertainty in our model predictions, cross-validation with the same data type, and then an independent data set for at least a subset of species.
Hey, right on. That's great. And Erica's also wondering if you considered habitat connectivity.
That is a good question. We discussed this early on. You can imagine that with 329 species, the number of predictors can get really large really quickly. And we struggled with connectivity to have an approach that would scale between species as small as a chorus frog or wood frog and something very large. And so we chose instead to focus on habitat amount and having a habitat variable related to patch size would at least get at not measuring connectivity per se, but evaluating sites that are part of a larger system of intact habitat. So indirectly, sense.
Yeah. And Aiden is wondering, which year of the AAFC annual crop inventory did you use, and will it be updated?
That's a good question. So the peer-reviewed paper and all the validation is based on a window with the 2015 AFC layer. And the idea was that we knew that that product would be updated through time. So instead of having to refit all the models, we could update the predictions with the new data. And so the public version of our biodiversity tool, the interactive map, those predictions have all been updated with the 2020 land use time series. Yeah.
Perfect.
And we will update it when we get 2025 eventually.
Okay, that's great. And Kim is wondering if you use data from iNaturalist at all, or only like if you use all data from iNaturalist or only research quality observations?
No, so we didn't pull from iNaturalist directly, but iNaturalist observations get deposited in GBIF, the Global Biodiversity Information Facility. So that's, if you think of a pipeline as data moves between different sets, GBIF is like the ultimate last holding storage for all of that data type. So from iNaturalist, it gets submitted to GBIF. With that data, we did an extensive amount of data cleaning so that it is research quality, but also a number of other checks related to, for example, the quality of the coordinate accuracy had to be high enough that we could assign that point to the size of the site we're measuring. If there was an uncertainty that's bigger than that, then we couldn't be certain that that species was observed in that exact site where we're trying to look at relationships to habitat. And so we had a large data cleaning pipeline that looked both at location accuracy, but also a number of other attributes, and they would have been limited to research quality.
Okay. And kind of along that topic, Brian from SAS Polytech is wondering, does the biodiversity map predictions account for sampling bias? For example, there's a lot more observations in the area surrounding Saskatoon than could be expected in a rural region, specifically like with iNaturalist. It's subject to this bias.
Yeah, that's a very astute point and something that we built into the modeling flow. So in that example, I showed the Great Plains showed like raw observations, and then background points that were kind of equally distributed. The way we accounted for spatial bias in search effort was to not just randomly sample background points. We sampled background points in a probability distribution that matched the distribution of effort based on where observations are. So in other words, we tried to mimic the density of points based on grouping all observations together. And so we did account for that, and we did some tests to see how well that represented it. And depending on the group, there's more or less spatial bias in where those species occur, but you can definitely see, rings around cities, for example, from just more effort in reporting.
Makes sense. And Catherine's wondering if you looked into any differences between the parkland area and the rest of the prairies. And Catherine says, I know that many of the highest species areas are associated with parkland moroso than elsewhere.
We did not build a predictor based on, for example, ecoregion, where we compared like parklands to the different grassland ecoregions, because we wanted to account for that basically in the type of land cover. So there's differences in climate, which would have been included as covariates, and there's also differences in land cover. For example, like, wetland density and total wetland cover is just much higher in the parklands versus some of the drier parts of the grassland ecoregions. So we didn't explicitly have an ecoregion predictor, but because of those differences being described both in soil characteristics, climate characteristics, and land cover, which we did include, we effectively were modeling for some of those differences. But we didn't actually go back and test whether richness is higher in one region versus another.
Nikki's wondering if your model would like the Northern Alberta boreal type of lands.
Sorry, I don't think I got the whole question there.
Nikki's wondering how this would work for the Northern Alberta boreal land base.
It's a good question. We've talked about expanding into different regions and what that might look like. And depending on the part of the country, there are different challenges around data availability and the type of land cover that's there. And so I think there's certainly opportunity, especially in the in the boreal transition area where there is like still quite a bit of observations to build models from, and similar land cover types where there's like similar agricultural land cover types where we can use a similar modeling framework to expand the model in that region. But there are also many parts of the boreal that are just really undersampled where it would be challenging to build these models, at least for some species.
That answer. Barbara's wondering, is it safe to assume that only native species are included?
That is a good question. We actually built models for some of the species that are naturalized, but they're not included in the predictions of relative richness. So we still built the models because they're species that are parts of those communities. Some of the species were eliminated because they were either transitional, if they just occur barely into the ecozone, or there was insufficient observations to build the models. But there are some non-native species that we did model, like widespread ones like house sparrow, for example.
Makes sense, yeah. And she's also wondering, are the observation data vetted?
Yes, they're vetted both from the sources that were contributing to us in terms of like they met a certain quality assurance from whoever was providing them or the source that we were requesting from. And then regardless of where they came from, we went through a secondary data quality assurance for some of those cleaning issues that I talked about earlier that were compared, for example, to like species range. There's some obvious issues around the uncertainty in location, but there's also errors related to the accuracy might be high, but it's outside the species expected range and habitat type where observations get cleaned out or flagged. And if we don't get verification, then they weren't included. So we did a secondary data clean on all observations. Yeah.
Right. Awesome. Thanks for that answer. We have another attendee who's wondering, why isn't time or season included in your model, especially for migratory species?
It's a good question. Time is an important factor affecting where species occur. Our interest was on general habitat suitability at a site. And so we were less interested in seasonal variation in habitat suitability, like is it suitable in this month versus another month? For birds, we only were essentially modeling breeding habitat suitability in that we limited the dates of observations to be during the breeding period. And so we didn't model overwintering habitat or migratory habitat. So in a way, that's underestimating the complete annual habitat suitability for a species, but we were most interested in habitats that support breeding populations.
Right. Fantastic. And we have a couple questions regarding AAFC. So Erica's wondering, how does this model compare to AAFC's wildlife habitat capacity on farmlands indicator? What are the differences? Are you familiar with that?
Yeah, I'm familiar with it. It works on a similar premise in that it's fitting habitat suitability to different land cover types and other variables. One of the major differences is the scale, where that tool works on the soil landscapes of Canada polygons. And so it's a coarser scale and they're not all equal size. The second is that they're based on expert and literature published associations between a species and a habitat, whereas we did not make an assumption of how species would respond to a specific habitat. We put the species observations in and the covariates in, and we let the model estimate how strong or weak that relationship is. And so it's a conceptual difference. I think they arrive at similar broad stroke conclusions about major drivers and differences in habitat suitability for species. But our framework also allows us to run some of those like tests where scenarios change the habitat suitability with a model framework that we have access to. Whereas I don't have access to the underlying habitat suitability of species to those land cover types for that capacity indicator, I don't have that. So that's my understanding of the broad stroke differences. But that one might be more in the weeds, and I'm happy to answer follow-up questions on that.
Okay, for sure. And there's another question already in from Noel. How was tame versus native grassland modeled with the AAFC land use categories? My understanding is that the data only has broad grassland and cropland categories. Do you have any thoughts on that?
Yeah, so there is there is a data quality issue where there's probably error in land cover, both in that category and probably several other categories, which is the nature of using remotely sensed data to assign land cover. The difference we used was around the separation of perennial cropland for tame grass, which is not done further back. And so you also couldn't do this with the earlier iterations of that data set. And we acknowledge that there's a bunch of efforts underway to try to better distinguish those two grassland types, but there's not a published product that is available at this scale that's open access that we could use. And so this was our best approximation, and then we will update as things become available.
Yeah, it's always the ongoing thing is land cover, and it's something that comes up at our PCAP meetings quite frequently is but yeah, we've got a couple other questions here. Aaron is wondering, is the publicly available data the same resolution, or is it coarser? For example, is it a quarter section scale?
Very, very good question. It is coarser. So the models are built on the quarter section scale. The public version is available in that mapping tool and on the data request form at a 4x4 mile grid square. And so part of that is around anonymizing specific parcels of land to assign some biodiversity to it. And also for some sensitive species where if given a specific place, even without the individual species identity, you might be able to derive which species occur there. And so it is at a coarser level. Part of that too is around our data sharing agreements to not share habitat suitability at that fine scale for individual species that are sensitive.
Absolutely. Aiden is wondering, is the model open source? Can it be run for one or a handful of species?
The full code is not currently open source. We can run, but the model, because of the way it's built on individual species models, we can run scenarios on individual species. And we have done that both for some internal tests and working with other conservation partners that have requested access to specific parts where we've done that.
Thank you. And Sarah's wondering if you are able to speak more to the ongoing work in Eastern Canada. Presumably, that area would have less agricultural land and more human settlement/roads.
Yeah, so that's a great point. The landscape is quite different. So some of the major differences that we're facing in that set of models is that there's much more species observation data. There are more species, and the patterns that are driving differences, especially in rural landscapes, are different. So in the prairies, where we were focused on this distinction between like perennial cropland, annual cropland, and then like grassland, that's not so much a major driver of patterns, but there is a much more dominant pattern based on forest type in Eastern Canada that we did some tests, for example, in the prairies of, distinguishing deciduous, coniferous, or mixed forest types and whether that added any benefit to us being able to predict which species occur where. And in the prairies, there was such low forest cover that it didn't actually make a predictive difference in terms of your accuracy because the overall cover was so low. In Eastern Canada, that's much different. And the communities are very different in some of those different forest types, and they're a larger overall percentage. There actually is quite a bit of agricultural cover. It's just a different agricultural type. But you're right in that there is a significantly larger footprint from urban and suburban development that's driving patterns. We're just at the stage where we're getting draft models for some of those species in that region, and that work's being led by a postdoc working with myself and Professor Julie Lee at the University of Ottawa, and Dr. Gisa Knathew is the postdoc leading that work.
Okay, and so they're looking at kind of correlations between like urban areas and species richness, or
They're basically an analogous approach that we took here, but some of the input data looks different because the variables that are driving differences are different for that community. And so we're still finalizing some of those predictor variables about like what should go in or out. But we know that the patterns are looking different already.
okay, interesting. And we have one other question here from another attendee. They were wondering if you think there's a correlation between natural habitat and a higher number of species, or why you think there's a higher biodiversity value around populated centers.
Yeah, that's a good question. So we spent a lot of time testing this when we were developing the models. And I'd say there's at least two things going on. One is that cities, especially in the prairies, are non-randomly placed in that they are around major rivers. And the river valleys are major drivers of, because you have multiple habitat types in a very small amount of area, you get these like multiple layers of communities where you have richer overall when you terrestrial vertebrate diversity. That's one of the patterns I think that's causing it. The second is that species richness is easy to interpret and understand, but is not the ultimate measure of like, it is not the only axis of diversity, and not like the be all end all of like, we should not just be maximizing the number of species. So as an example, like a really good example is the diversity of vertebrate species that occur in native grasslands is just much lower than a lot of the other habitat types in this system. That doesn't mean that that's a very unique set of species, but the total number of species is lower. And so using richness as the only metric is not the be all and end all. And so there might be more species around some of those cities, but it doesn't mean that it's capturing all of the variation in biodiversity that we should be conserving. And so the species identity matters, I guess, is the take home of that.
Okay, that makes sense. Very interesting about your comment that cities were not randomly placed. You're absolutely right. That makes sense. So how long have you personally been involved in working with this?
Several years. I think we started in 2021 was when we first started work on this project, and our paper came out last summer. And so yeah,
Fantastic.
it's been a
And
process.
I bet. And so what are your goals? Like, what's your vision for the future use of this tool?
I think it's to help do better conservation action, and to put numbers to our work beyond just measuring area conserved, to understand which species are benefiting where, and to be able to stretch, our dollars further to have the largest impact we can.
Absolutely. That's really awesome. I can see that's be really useful for a lot of conservation organizations. Yeah. And it's really nice that the tool is available to the public to see. Have you had a lot of feedback from the public about it?
We just launched a few months ago. We've had some data requests come, and we're just starting to get feedback on how people are using it. But if you are exploring and you see a use or a way you'd like to see it improved, we'd be happy to hear from you. There's a contact form on the tool website too for you to get in touch. And I think my email was on the last slide as well.
Yes, yeah. I've personally checked it out, and I just thought it was really neat to be able to see that, the area I live in is so rich. Or I go to the city of Swift Current frequently, and I was amazed at the richness of that city there. I'm glad that you're able to explain again, the Swift Current, the river valley that's there too. But yeah, that's all the questions that we have from our attendees right now. So I just want to say thank you so much, James, for this really amazing presentation. It's really detailed and kind of walking us through the background behind this tool. It's clearly been a passion of yours for a number of years. So thank you for sharing that with us today.
Thanks so much for having me, and I hope to hear from folks that are using it and how we can make it better for your use case. So thanks again.
Absolutely. Yeah, I think it's going to be a really, really useful tool. To all of our attendees, thank you so much for catching today's webinar. As I mentioned earlier, our webinar is being recorded and we will put it on our YouTube channel in the very near future. So check that out if you missed anything or if you want to share it to somebody you think will be able to use it in the future. And also check out our PCAP website. We have a really awesome lineup of webinars coming up this winter. So you'll be able to register for those, and there's more being added all the time. So check that out. And when you leave today's webinar, there'll be a quick 1-minute survey. So if you don't mind filling that out, we really appreciate hearing back from everyone. And it'll keep our new speaker series going into the future. So with that, thank you so much, everybody, and have a great rest of your day. Bye now.